Okay, so we are going to pick back up with the digestive system, and this is starting with the small intestine. So we've been all through the stomach, and we talked about structures related to that. The next organ that we're going to come to as we travel along the digestive tract is going to be the small intestines, and this is the major part. It plays a major role in digestion and absorption of water and nutrients. So a lot of activities are happening here in the small intestines. Our small intestines are about 20 feet long, so that gives you an idea of how long they are. And what we see actually with our intestines, both small and large, is in the cadavers, they tend to be a lot longer. We could stretch them out even more just because we don't have muscle tone in cadavers that we would have in a living person. So in a living person, intestines are about 20 feet long. And the small intestines are going to run from the end of the stomach, which is going to be signified by the pyloric sphincter. So if we have, here's our stomach. And if you recall, the pyloric sphincter was this little thickening of smooth muscle at the end of the stomach. And then we start into small intestines. And small intestines are going to run from that pyloric sphincter all the way to a sphincter at the other end, and that's called the ileocecal valve. And so what we're going to see is actually the small intestines are divided into three different individual parts. So the first part is the duodenum, which would be this part here. Maybe we'll just put a D by that. The first part of the small intestines as we leave the stomach is the duodenum. The next part, the middle part of the small intestines, is going to be the jejunum. So we'll say this is kind of our middle part. And then the third part of our small intestines is going to be the ileum. So if we continue along, we're going to end up with the ileum, which is going to be our most distal part of the small intestines. And what we have at the end of the ileum here is another sphincter. In this case, we call it a valve, but it does the same thing because it's going to control flow going from ileum into cecum, which is the beginning part of the large intestines. And so here we would start kind of large intestines coming up and around and down the other side, and we'll talk about that a little bit more, but this connection right here is the location of our second valve. And so this is going to be our ileocecal valve, signifying the end of the small intestines. So beginning at the pyloric sphincter at the junction of the stomach and the duodenum, small intestines, and the end of the small intestines signified here by the ileocecal valve, which is a sphincter controlling flow going from ileum into cecum, which is the beginning of the large intestines. Okay, so arterial blood supplies the small intestines through the mesenteric arteries, and so we know primarily that's going to be superior mesenteric because it does small intestines. And in terms of venous drainage, we have the superior mesenteric vein that's going to drain the small intestines, and because it's a digestive organ, it's going to get routed into the hepatic portal vein and back to the liver. So small intestines does have both sympathetic and parasympathetic innervation. So we know that at rest, because rest and digest, our digestive processes are going to be stimulated. So in that case, our parasympathetic input would be increased and sympathetic input would be decreased. If our sympathetic branch of the autonomic nervous system was the predominant force at that point in time, then we know that digestive processes in the small intestines would be minimized and inhibited because of that. Okay, so here's your picture of the small intestines. Just by way of review, here's esophagus coming on down. We're going into stomach, and then this right here would be the location of the pyloric sphincter. So controlling flow going from stomach into duodenum. We go through the duodenum first, then we have jejunum through here, and then we have ileum, and then we're finally going to end ileum as we transition into the large intestines. Okay, there's a few things that happen in the small intestines, a few adaptations that are present in order for the small intestines to do what they need to do. And so their primary role is going to be nutrient absorption. So there's a few things that are going on to allow this process to happen. So one of these things is the presence of what's called plica circularis. And so what this is if you cut open the intestines and you look at it from the inside, you've got these kind of circular folds on the inside of the intestines. They're not smooth. They've got folds and ridges, and they're kind of circularly arranged along the length of the intestines. And what this does, if you look here, we've got a cross-section of the small intestines, and you can see these kind of circular folds. So if you kind of open it up, you would see these circular folds on the inside. And what that does is it basically increases surface area and allows for more absorption because of that increased surface area. And it also slows down the movement of chyme moving from the stomach into the small intestines to give it time for reabsorption of nutrients. We've got a second adaptation, and these are going to basically be the presence of villi. And so these are little finger-like projections that come up from the surface, the interior surface of the wall of the intestine. And so what this does is that you have capillary beds, these small capillary beds that are located all around the intestines and specifically around these villi. And so they are there for absorption and can increase surface area and allow nutrients to be absorbed into them and then transition right into the capillary bed. And then the third adaptation that's going to be present is microvilli. And so what happens here is that you have, we call this a brush border. So on the very inside of the intestines, you're going to see this, it kind of looks fuzzy. These microvilli create what's called a brush border, and they are the location for enzymes that are membrane bound. So meaning that they stay right there, and you have these enzymes that are present, and they're going to help with digestion of carbs and protein in the intestines. Okay. So a couple of different pictures here. This is a cross-section of the intestines, and you can see the plica circularis that are going to be the folds in the wall. Here's your villi. So these are going to be the finger-like projections that project into, kind of here, this is a nice one too, project into the lumen of the intestines that allow for increased absorption to take place. Okay. So when we talk specifically about what's happening in the small intestines, what digestive processes happen here? So again, this goes back to that whole list at the very beginning and what process is happening at which location as we're going along the route. So what's happening in the small intestines? We do have propulsion happening. So we have this movement called peristalsis. So rhythmic contraction and relaxation of segments of the intestines to help move food along the GI tract. We do have mechanical breakdown. And so with the smooth muscle contractions, we are kind of pulverizing the food that's contained inside the intestines. And that also helps to kind of mix up the food with the enzymes and the bile to help increase their access to the food, and it helps to speed up the process of digestion. So we also have digestion that's happening in the small intestines. So we know that pancreatic juices contain lots of different enzymes, and we also have enzymes present, those membrane-bound enzymes in the brush border along the interior of the small intestines that help us to digest all types of food that we eat. And we also have the process of absorption that's happening in the small intestines. So carbs and proteins and fat, and we're reabsorbing water. So all of those processes are happening in the small intestines. That is where the most of our absorption is happening for all of our nutrients. Okay, so for all of these guys. If you remember from stomach was not a hotbed of absorption, right? There wasn't a lot going on there. It was only alcohol and aspirin. So most of our nutrients are not absorbed in our stomach. Most of our nutrients are absorbed in our small intestines. So, okay, which brings us to our next large organ, and this is going to be our large intestines. So we can also call this our colon. So you'll see that used somewhat interchangeably. Large intestines, as you would possibly suspect, have a larger diameter than the small intestines. They're not quite as long though. In a living person, it's about 5 feet of large intestines. And we've got some different things here. So let me just point out some of these. On this picture here, we're seeing at this area here the end of the small intestine. So this is going to be the ileum, and this is going to be the beginning of the large intestine here. Okay, so this is the location of that ileocecal valve. And if you remember back, we talked back in the beginning about a hormone called gastrin, and its release prompts the relaxation of the ileocecal valve. Okay, and so that's what's going to allow movement from the small intestines into the large intestines to finish out its process. Okay, so that's ileocecal valve. This beginning part of the large intestines is called the cecum, and this is basically like this little pouch here at the bottom. Okay, what's important in this area is that your appendix is located here, and that's going to be this little guy right here. And we'll talk about your appendix in just a second, but your appendix is hanging off of your cecum. And then we start going through the rest of the large intestines. So we have the ascending colon. So we're going to have large intestines that basically travel up. So we're traveling up this way. Then we're going to take a turn at the hepatic flexure. And if you remember, what does hepatic mean? What do we think of when we see the word hepatic? So it's liver, right? And so if we were to draw, our liver would be about here, So that's why we call this the hepatic flexure. We're going to take a turn, and then the large intestines are going to cut across the body, and we call this the transverse colon, okay, because it's kind of in the transverse plane. And then they're going to take another turn, and so this turn is called the splenic flexure. So we kind of have this turn here where we start coming back down the other side. Splenic flexure, because on this side, on the left side of the body wall, we know is the spleen, right? So large intestines are going to travel back down the other side, and we call this the descending colon. They're going to travel all the way down. They come to this little S-shaped curve here, and we call that sigmoid colon. And then at the very end, we have the rectum, which is going to be kind of this distal end. And then at the very end of the large intestines, we have the anal canal, which is going to be right here at the bottom. And that's going to open it up to the external world, because that's the other end of our GI tract. So just some anatomy, and we're going to review that in lab too. I do want to point out a couple other things. This Tinea coli. If you look at this image, actually let me see if I can get rid of all of that. There we go. Okay, so we'll kind of start over again. This Tinea coli, oops. Okay, if you look at this image, we've got this kind of this band here, right? It's coming up the middle of the large intestines. And so what this is this is a band of smooth muscle that basically kind of cinches up the large intestines, and it kind of like creates these pockets of intestines. So if you would picture it nice and smooth, if we didn't have this structure there, but that Tinea coli, it's kind of a band of smooth muscle that runs across or along the large intestines, and it cinches up the large intestines. So it kind of pulls it, and what it does is it creates these pockets of large intestines, and we call that haustra or haustrum. So it kind of takes the large intestines and it creates like these kind of balloons of large intestines, you know, inside them. And then we also have this other structure called epiploic appendages. And so what these are, if you look at this image here, you see all these little yellow, these little like, they basically look like just little fat droplets hanging off of the large intestines. These are called epiploic appendages. And they're just little kind of droplets of peritoneum and fatty tissue, and I'm not aware of any functional use for them. They're just there. They're normally there, but I'm honestly not sure what they do. But you see them. They're very identifiable when we see them in cadaver lab. So just some things related to large intestines. So here again, here's our large intestines. I did want to point out the appendix, little appendix hanging down off of the cecum, which is the first section of the large intestines. And then we've got ascending colon, transverse colon, descending colon, sigmoid, rectum, and then anus. So those are the different divisions of the large intestines. So a little bit more about appendix, just because that's interesting. And like we said, it's attached to the cecum. And so what the appendix is it's a little collection of lymphoid tissue. So it plays a role in lymphatics. It's actually part of this collection of tissue called MALT, which is mucosal associated lymphoid tissue. And we see these collections of lymphoid tissues all through the digestive tract. And so this is just one example of them. And what happens is the appendix plays a role in immunity, and it also helps to recolonize the bacteria that is naturally present in your gut. But like with everything else, it's very interesting when things with the appendix go wrong. And so what can happen is feces can actually end up blocking the entrance to the appendix and trap bacteria inside it. And you can end up with a pretty serious infection of appendicitis where they would have to go in, hopefully endoscopically with little tiny incision and take your appendix out. So if any of you have had that done, you've had that experience. But that's what happens with the appendix. So what happens in the large intestines? What's the function of the large intestine? Well, food, it's kind of routing through. It typically spends anywhere from 12 to 24 hours traveling through the large intestines. The main thing that's happening in the large intestines is reabsorption of water. So basically at this point, we've reabsorbed all of the nutrients because we did that in the small intestines. So we've got all of the nutrients that our body needs that we've already reabsorbed. And at this point, we're basically just reabsorbing as much water as we can. So the rest of what we can't reabsorb or don't reabsorb at this point, we are going to eliminate as feces. Okay? So in terms of histology of the large intestines, there are no plica circularis here. There's no villi here because the food has already been absorbed prior to getting here. We don't need it in the large intestines. What we do have, though, is another very thick mucosal layer with goblet cells that are responsible for producing mucus, because all that feces needs a little help in order to be excreted from our body. And so that's what the mucus in the large intestines is going to help us do. Okay, so here's your digestive processes broken down for the large intestines. We do have propulsion that's happening here, and those haustra, kind of those balloon type areas in the large intestines, when they fill with food, it causes them to contract. So it's a reflex that happens automatically. So when they're filled up with food, it actually causes the smooth muscle to contract, and that helps propel the food along our large intestines. In the large intestines, we do have minimal digestion happening. So there's really not a lot of reabsorption of nutrients because we've already had that happen, but we do have bacteria that's naturally present. And so that is a process that's happening in the large intestines. We do have reabsorption or absorption of water and salt. So we're able to pull back in as much water as we can so that we don't get dehydrated. And then our final process in the large intestines is defecation. And so propulsion is going to take care of moving all of our food or the residue of our food all the way down to the rectum and then eventually to the anus. And what we have when that food residue gets to the rectum, it's going to distend the rectal wall. It kind of stretches it, and we've got receptors that detect that stretch, and that's going to initiate what we call the defecation reflex. And so this is going to activate the parasympathetic nervous system, which we know parasympathetic is rest and digest, and part of digestion is defecation. So it's going to lead to that process happening. And this is what we have here. A couple things that I just want to point out on this. Here's the end of our large intestine. So here's our sigmoid colon, and then we head down to the rectum, and we do have anal sphincters that are located in the walls of the rectum. And so we've got first an internal anal sphincter, which is going to be this kind of more internal muscle through here. And this one, an important difference here is that our internal anal sphincter is made out of smooth muscle. Okay, it's smooth muscle. It is not under voluntary control. Smooth muscle, it is involuntary, and it is part of the parasympathetic nervous system. So when you have feces that are moving into the rectum and they're distending the walls of the rectum, it's going to activate these little sensory receptors within the wall of the rectum. We're going to send that signal to the brain, and that's going to activate the parasympathetic nervous system, which is going to travel down. It innervates the smooth muscle. That's part of the internal anal sphincter, and it's going to tell it to relax. Okay. Now there's a part 2 of that, thank goodness, because we want to have some control over this, right? So we have some control. We have an external anal sphincter, which is comprised of skeletal muscle. And so that's going to be all of this muscle out here on the outside because it's external anal sphincter. And it's skeletal, so that means it's voluntary. So we do have some voluntary control over this. So we have voluntary innervation to that skeletal muscle. So if we get the signal from our parasympathetic nervous system to say, hey, defecation is happening, if it's a convenient time, then we have control over our skeletal muscle to say, okay, yes, we will allow our external anal sphincter to relax, and that's going to let the feces out of the body and complete the whole process. Okay. And then really the last thing that I want to talk about, I just pulled out some big picture concepts here on processing of a few different nutrients that we ingest. So one of them, this first one, is going to be carbohydrate processing. And what I'd like to point out here is, so in this image, this is just to orient you, this is the lumen of the intestine. So this is the inside of the intestine. This is going to be the apical membrane, because if you remember, cells have an apical surface and a basal surface. So this surface here of the cell is going to be apical because it's going to be open. Maybe these would be the cells on the other side of the intestines, and so this would be the lumen of the intestines, right? So the apical surface of the cell faces the interior of the lumen, and the basilar or basolateral surface of the cell is going to be anchored at the bottom, typically, on a basement membrane. And then, as we know, we've got capillary beds that are going to be, highly numerous in these areas, because anything that we absorb from the lumen is going to pass through these cells and right into our capillaries to travel throughout our body. So that's the general setup. So what do we have with carbohydrate processing? We have amylase, which is going to be the enzyme that breaks down starches, and it's secreted as part of our pancreatic juice that we talked about with the pancreas. So pancreatic amylase is going to break down starches and glycogen into smaller oligosaccharides and disaccharides. Those are going to come into contact with brush border enzymes. And so these are going to be located on the villi or microvilli within the walls of the intestines. And remember, they're membrane bound. So those enzymes are basically just hanging out there, and they're going to end up breaking them down into monosaccharides. So we're breaking them down into smaller pieces that our body can use. At that point, those monosaccharides are going to be transported into the cell across this apical border by co-transport with sodium. So sodium is going to enter down its concentration gradient, and it's actually going to pull these monosaccharides with it. Okay? Once it's in the cell, it's then going to travel through the cell that creates the wall of the intestine, and we need to get it into the bloodstream. And so those monosaccharides are actually going to travel through facilitated diffusion, through a carrier, a membrane-bound protein carrier that's specific to those monosaccharides, through that basolateral membrane, and then they're going to come right on into the capillary, because as we know, capillaries are porous, and we have different types of capillaries, and what we see in the digestive tract are going to be increasingly porous capillary beds that help with absorption. And so they are going to be fenestrated capillaries that allow substances to pass through. And that's exactly what we're seeing here. So they're going to enter the capillary bed. Okay. We also have protein processing. So what's happening here? So again, this is our pancreatic juice that's playing a role, and proteases are the class of enzymes that are responsible for breaking down proteins into smaller proteins, and then actually even amino acids. And that's what we're going to see here. Those proteins, smaller protein fragments, and even amino acids are then going to come into contact with the brush border enzymes that are present again and further break down those smaller proteins into amino acids. We travel here. Those amino acids are going to be co-transported across the apical membrane with sodium. So you see a trend here, right? Sodium is our primary co-transporter in this scenario. And it's basically going to travel across the intestinal cell. And then we have a membrane-bound protein transporter that's going to carry our amino acid across the basolateral membrane of the cell, and that's going to happen through facilitated diffusion. So those amino acids are going to travel down their concentration gradient, but it's facilitated diffusion, so it does need a carrier in order for that to happen. And then those amino acids are going to travel right in between that intercellular cleft and right on into the capillary. Okay, and then the third one is fat processing. So how do we deal with lipids? And so basically what we're seeing here is kind of the highlight of this is emulsification, which we talked about before, where we have these large fat droplets that come into contact with bile salts from the liver and the gallbladder, and it emulsifies them. So it breaks them down into smaller fat droplets, which increases the surface area so that the enzymes can have a larger space to work. So again, we have enzymes that are present in the pancreatic juice, and in this case, because it's fat, our enzymes are going to be lipases. And so the lipases are going to break down or hydrolyze the triglycerides into smaller monoglycerides and free fatty acids. Those are going to end up joining and creating what's called a micelle. And so these are basically just tiny little, they're even smaller than like the fat droplets. They're just tiny little kind of specks of fat and fatty acids and monoglycerides. So they break down. And these are small enough now that they can actually diffuse through and enter into the cells that are lining the wall of the small intestines. Okay, so they're going to diffuse through. Now, what's interesting is that the micelles end up combining with other fatty substances and even proteins that are in that area to form what are called chylomicrons. Now, what happens with the chylomicrons is that they are actually too big to diffuse into blood vessels. Okay, so they are actually going to enter the lymphatic system initially, and then the lymphatic system ultimately drains into the venous circulatory system. So they ultimately end up in the bloodstream, but initially they're too big, and so they have to drain into the lymphatic system first, and then they drain into the blood eventually.